Hello everyone, and thank you for inviting me. I'm very excited today to present you the importance of virtual human twins in cancer. And so I have the not some simple mission of explaining to you in about 30 minutes what's cancer, because I assume that not all of you are familiar with cancer, with oncology, the specialty that treats cancers, what's complex about cancer, and why we believe that we need tools like AI modeling and virtual twins as physicians to be better, to bring better treatments for our patients. So I'm Julien Vibert. I'm working in Gustave Roussy in this large anti-cancer center in Villejuif. I'm in the drug development department, and I have a background in machine learning and genomics. And so today I'll present you first cancer. So if you're not familiar with this, cancer is a disease that involves some hallmarks that were described about 20 years ago. So you have a cell that will become cancerous when it has some characteristics that allow it to grow. So you see, you have 6 hallmarks here, and the most important probably are the fact that you have proliferative capacity of the cell that is abnormal and that is sustained, and you have what is called replicative immortality that you see in the bottom, but you have also other properties of the cancer cell that will make it resist cell death, but also attract new vessels to feed it. So all of this leads to a cancer because the tumor grows, and of course it leads at one point to the death of the patient, unfortunately. So the understanding of cancer has seen exponential progress, I would say, in the collection of data, but also in the theories that were proposed to explain cancer. And you see that these hallmarks of cancer was further progressing in 2011 to having more properties that were described. And I will not detail all of them, but some of them are really important to understand, is that, for example, you have genome instability in the bottom left, meaning that in a cancer cell, its genome can be unstable and acquire new mutations. And so you have this very dynamic nature of the cancer cell that will evolve across time, but also evolve with the treatments that you give to the patient. And so you have this very dynamical nature of cancer that makes it really hard to treat. You have also, for the first time, some hallmarks that involved not the cancer cell, but in fact its microenvironment. For example, on the top right, the immune system. You know that immunotherapy has become in the last years some very effective treatments, and it's all about the immune system. And we know that the immune system is avoided by cancer cells, and we have to make the immune system recognize the cancer cell so that it can destroy effectively the cancer cell. So I will not detail all of this, but just to tell you that 10 years after, and this is currently probably the latest global view of the cancer cell, we have 4 other hallmarks that were added to the theory of the hallmarks that you see on the right, and some of them are really interesting. You see on the top left, phenotypic plasticity, meaning that the cell once again is very dynamic in nature, and in fact we have this whole field of epigenetics that you see on the top right, which involves all the changes that are not directly on the DNA sequence, but that are heritable across cells in a somatic way, and that will let the cell grow and adapt to its environment and to the treatment that is given to the patient. And finally, on the bottom right, you have the microbiota, which is becoming a hallmark. And once again, it's not about the tumor cell, it's about its environment, and especially all the bacteria, because you all know that in the human body, most of our cells are in fact bacteria. And so the microbiota influences the development and the treatment of cancer. This latest paper this year was about also introducing other dimensions that we take into account when we treat a cancer as a physician, as an oncologist. In fact, we have to take care about the patient in its globality. You see the clouds that are over those hallmarks. Some of them are about diet, some of them are about exercise. And you all know, and we all know as physicians, that these are very important determinants of how the patient will be evolving during treatment. And also, I don't think it's here, but psychology of the patient is really important. And so as oncologists, we try in our little brains to integrate all of this to find the best course of treatment for our patient. So I hope that you see now that cancer is really a complex disease, is a systemic disease that we have to consider as a whole, and this makes it really difficult to treat. But of course, with all this understanding, we think that we have more tools now to better treat cancers, and indeed we have, I think, improved the survival of some of our patients, unfortunately not all of them, and our objective, of course, is to go further. So cancer as a systemic disease, you have all about cancer cells, but also about its microenvironment, so all the cells that you see on the left that are in crosstalk with the tumor. You have all the other organs, especially that will be metastasis. So when a tumor cell goes in another organ, it's called a metastasis, and so it depends on which organs will allow which type of tumor to grow inside itself. You have, of course, the systemic parameters. Cachexia is inflammation or weight loss because of malnutrition. So all of this we have to take into account, but also some very dynamic macro factors that you see in the bottom, like diet, exercise, microbiota, etc. So how do we treat cancers now that we know all of this? Well, we are currently in the era that we call precision medicine, precision oncology, especially in Gustave Roussy. So you see on the left what was currently, or at least in the previous years, done, meaning that for all patients with the same type of cancer, you would give the same type of chemotherapy. Usually it was chemotherapy. And with all the research, the trials that were done in the previous years, we are currently moving towards this right part of future precision medicine, which is more about personalized treatment. So how do we do this? In fact, you have seen all the determinants that are involved in cancer, and a lot of them are in fact molecular or cellular, and so fortunately we have seen many progresses in technologies that allow us today to assay all of these molecular changes at the DNA level, but also at the RNA level, the proteomic level, on individual patients. Of course, we need to go and biopsy, meaning to take a tissue sample of the tumor, but this we do. I mean, in Gustave Roussy, we are doing a lot of biopsies on our patients. We are sequencing the DNA, for example, and this allows us to have very personalized diagnostics for patients, and as you will see, we have also targeted therapies that we can give based on these molecular characteristics. So this was a review in 2015 showing what was the paradigm at the time, saying, okay, oncology will be, we want to identify the mechanisms in each individual patient and not anymore do treatment for one cancer type for all the population and say, okay, this treatment is working statistically better than another treatment. It's all about population level. Right now, we want to do personalized medicine. You saw all the determinants. Clearly, I would say that cancer is N of one disease. Each cancer, each patient is different. And so we want to be able to characterize each patient, each tumor, and give it the best treatment at the right time. This was historically one of the first papers. So Fabrice André, my boss in Gustave Roussy, who's one of the leaders of this precision oncology initiative a few years ago, really advocated for this kind of medicine where you do molecular screening for patients. So you really want to go deep into the molecular diagnosis of the patient and propose, based on the molecular alterations at the DNA level, targeted therapy. So orient them towards, for example, clinical trials A, B, or C, and really test the power of those targeted therapies based on molecular screening. And in fact, we think that this vision was correct because it led to real improvements for patients. Here is a paper that we published in Gustave Roussy in 2019 about the genomic characterization of breast cancers. You don't need to read all of this, but basically the message is that a lot of those tumors have recurrent mutations that are called driver alterations, meaning that there is a mutation in a gene, oncogene or tumor suppressor, that will lead to the development of cancer. And of course, you want to be able to target this driver alteration. Unfortunately, a lot of those driver alterations are in fact not recurrent in patients. They are rare. They are in a subset of patients. And of course, you want to be able to give the best treatment to the right patient. And if all of them have different drivers, you have to have a drug for each different driver. But still, we had some alterations like this, for example, the P10 mutation, where it's rare as a subset, but you have a very effective therapy. And that's the paradigm that we want to follow, is that even though the alteration is rare, we want to identify it, we want to find the right treatment for this alteration and be able, as you see in the bottom, to have the tumor regress for the patient. So there was a lot of development about genomic characterization of tumors, characterizing the driver alterations and trying with clinical trials with products from the pharma industry targeted to specific molecular alterations to find the best treatment for each molecular alteration. You see, this was a seminal paper in 2018, also with Gustave Roussy, about developing guidelines for oncologists. This is the European Society of Medical Oncology, ESMO guidelines, and we have some recommendations for molecular targets. For example, class 1 molecular alteration means that for this type of alteration, you have potentially a drug which is either on the market or on a clinical trial that can be used and that will potentially improve the survival of the patient. And access to this treatment should be given in priority. Whereas for evidence tier 2, this is a molecular alteration where we have fewer evidence to recommend this drug, but still it means that you have this framework about molecular alterations and corresponding treatments that you can potentially give to patients. And what was really interesting is that based on this framework, we did this in Gustave Roussy, so that was published in Nature 2 years ago. We stratified patients based on their molecular alterations, and we gave treatment based on this molecular alteration. On the left, either if it was SCAT really 1, like really important evidence to say that this will lead to improved survival, and in fact this was the case. You see the survival curves are better in the targeted therapy, whereas when you have less evidence, it's not so clear that you improve survival. So in the end, is to tell you that we have really in cancer right now a lot of research that is done about finding the right treatment for the right molecular alterations. And this is really important, especially for rare cancers. As you saw, a lot of cancers have rare driver alterations, and we have to be able to develop drugs and to validate drugs for those patients without being able to run big phase 3 trials like in cardiology. We don't have thousands of patients with a rare cancer. Sometimes we have like 10 patients each year in Gustave Roussy for one type of cancer. So how do you develop trials for this? Of course, and you see me coming, being able to do synthetic arms for virtual trials would be very important, I think, for these rare diseases, and to validate those drugs and registrate those drugs to the FDA, for example. And this is the vision that we are now coming to. So I will go more briefly on this, just to present some of the things that we do in PRISM, which is the IHU that is developed in Gustave Roussy for molecular medicine in oncology. And our vision is really to have a lot of cutting-edge projects with technologies, with patients, with a lot of samples to classify cancers really based on biology at the individual scale. Once more, the key word is being able to do it on the individual scale. And of course, we want to be able to decrease mortality, burden of the treatments, toxicity, and all of this should be done, I think, in the next years with all those projects that we are launching in Gustave Roussy and all over the world. I will give you just some few examples of about what we can do right now as cutting edge, I would say, technologies. You see here the clinical utility of circulating tumor DNA, meaning that usually when you want to assay a tumor, you have to do a biopsy, a tissue biopsy. So this is very invasive, it can be dangerous, but now we have tools that are able on a single blood test to be able to detect the circulating tumor DNA. And this is based on a blood test. We can detect if there is tumor DNA in the blood, and you see here on the curves that the patients that will have circulating tumor DNA detected will have worse survival, and in fact this is a feature that we don't have in clinical practice. But if you are able, as you see, to have this feature and say, okay, this patient in fact has circulating cells that we are not able to see with other tools, you want to change treatment early for these patients. So this is the kind of tool that we are developing. The other kind of innovative tools is organoids. Organoids are 3D models of a cancer. So it means that you can extract a cancer cell of a patient, put it into culture, and not classical cell culture where you have 2D growth, and it's really like, it's not a real good model, I would say, of the 3D organization of the cancer in a patient. And organoids allow this, and we have a specific culture media that can allow us to do this at a patient level. And so the vision that we have in Gustave Roussy is, for example, this clinical trial where we develop for individual patients organoids on which we test different drugs, and then we give to the patient the best drug based on this testing on this organoid model. And our hypothesis, of course, is that this can lead to personalized medicine based on this model. This unlock program is another thing, and it's to insist on the dynamic nature of cancer. When you do a biopsy, when you do a treatment right now, you will base all of your reasoning on a biopsy that was done at the diagnosis. But in fact, cancer, as I showed you, evolves. There are epigenetic alterations. There is the immune system that will remodel the tumor. And if you are not able to sequentially assess the tumor, it's probably and it's surely a problem. And so the Unlock program is about biopsying patients sequentially, and so mapping the different alterations across time and being able to do this kind of modeling across time and better extract what will be important for the oncologist. And finally, another kind of innovative project is about AI. I will come to it. So we are very involved in oncology about the diagnosis on pathology slides. This is our gold standard to say, okay, this is this type of cancer. And of course, AI and deep learning especially is very good at analyzing this kind of images and classifying in the right type of cancer. And even, as you see in this project of Gustave Roussy, predicting survival based on characteristics, morphological characteristics of the pathology slides. So to finish on this, to tell you that in PRISM, but also all across the world, we are currently developing novel technologies to go further into the multiple scales of cancer, molecular, but also organoids, it's more macroscopical, spatial transcriptomics. We have all of these innovative technologies that we want to use for each individual patient to better predict the individual nature of the tumor and how we can treat it better. We do it in Gustave Roussy, we do it right now all over the world. And I would like to finish on why do we need AI, why do we need model. I think you all know now that it's it's necessary with all the data, the complexity that I showed you, that we need to be able to treat cancer. We need to aggregate and integrate multiple scales of data that you see here on the left, what we call the omics, genomics, transcriptomics, proteomics, epigenetics, but also imaging, pathology, and all the other social determinants. We have to integrate all of this, the electronic health records. We have to aggregate them across time, across cohorts, across hospitals. There are lots of issues about standardization of data. This is a nightmare, but we have to do this to better understand cancer and better treat patients all around the world. We have right now a lot of AI studies in cancer research. You see on the top here, it goes from basic research to translational to clinical research. All over the spectrum of research, we can use AI tools that are based on all these data that we collect on patients and that we want to use to predict the best treatment for our patients. Currently, we have these 4 types of data mainly that we use for AI. These are very rich data, image data like radiology, but also pathology I showed you. We have, of course, genomics, large databases of sequences of DNA, RNA, but also a lot of EHRs, so natural language and large language models are becoming also very useful tools for us oncologists. So to come down to modeling, this is more theoretical, I would say, and for us oncologists, it's not necessarily very clear, but we know that we can do bottom-up or top-down modeling. Usually we were more about bottom-up in the sense that we liked as oncologists to go inside the signaling pathways to say, okay, this molecular alteration leads to this alteration in this signaling pathway, so we are going to give this drug that will block this pathway. And right now with AI, all the data that we have, we're more about probably the top-down approach in the sense that we want, as you said very nicely before, we have a lot of data that we can use with AI to have this very large multi-scale approach, even if it's not necessarily, sometimes it's black box, and I agree with you that we need mathematics, we need modeling to better understand, and also for the physician it's really important that this is not a black box. Of course, our objective is to go towards generalist medical AI in the sense that for now we are mainly developing AI models for one type of modality, pathology or radiology or genomics. Right now we want to integrate all of this. We want to reproduce, in fact, what our little brains of oncologists are doing, so integrating all of this and trying to find the best treatment based on our experience. And of course, AI can learn a lot of experience, learn a lot in the clinical literature, the research literature, and of course, generalist medical AI is like what we want to do with all of this. This is an example of a project that we know we collaborate with Gustave Roussy. It's about building a foundation model based on all this data that I showed you and being able to use it as a kind of generalist foundation model for oncology. I will skip that, but basically this is a nice paper that shows that integrating pathology, EHR, imaging can lead to a good prediction to response to immunotherapy that was shown in a recent paper. Just one word on generative AI. We are very excited about this, not only because we are talking and we want to have synthetic arms for clinical trials, but for rare diseases, rare cancers, we are very excited about potentially generating some synthetic data, including biologic data. So there are many debates about is it real, like real data that we can use for research, but this is a very promising tool as physicians that we want to explore. So I will not delve on this because you had many talks already on this, on synthetic control arms, but I think this is really a major advancement that we are expecting as physicians, especially as carers of rare cancers. I personally treat sarcomas. You may never have known or heard about sarcomas. These are very rare cancers of bone and soft tissue, and we are really in need of this because basically we give chemotherapy to everyone and it doesn't go very well usually. So why do I tell you all about this? It's because, of course, I want to introduce the concept of the cancer patient digital twin. This is a concept that already in Nature Medicine 3 years ago was alluded to in this paper. So it's all about creating a twin, digital twin, virtual twin for cancer patient. I say patient because, of course, there is the twin of the tumor, but as you saw, there's not only the tumor, there's the microenvironment, the immune system, the microbiota, and all the determinants of the patient that will make it a unique patient that you want to be able to model, to simulate, and to treat the best way. So you want to be able to integrate all of the determinants of your patient with the experience that you have, but also of the literature, so all the clinical trials, the clinical recommendations of the experts. So to give you some examples, these are not necessarily twins of a patient, but there was this paper this year about creating a molecular twin for pancreatic cancer, which is one of the most deadly cancers. So it was about characterizing in depth a lot of patients, hundreds, with very cutting-edge omics technologies, including genomics, transcriptomics, and being able, based on all of these markers, these high-dimensional data, to develop AI models to predict survival, but also discover novel targets for treatments. And this worked quite well. There is this other, more ambitious project, I would say, it's still a project. It's about building a virtual child. So it was presented in Cancer Discovery this year. It's about a large consortium, including industry, to try to, of course, collect a lot of data to be able to model a virtual child. And of course, childhood cancers are rare. They need a lot of research. We are not able to do large clinical trials in pediatric oncology, and this is the kind of project that should be transformative for patients, for children with cancers. So to finish, what do we want to do if we want to create a digital twin for cancer, for patients? This is the objective of the MediTwin project that we are involved in Gustave Roussy. So on one part, of course, we want to be able to build a digital twin based on all the data that I presented you, not only molecular, but also EHR and environmental data. We talked about real-world sensors. There is this dynamic nature of the twin that we want to be able to update in time. We have this concept of knowledge cards, which is about the fact that this twin, in fact, so there is the twin of the patient, but we want to have a tool that is also, that has experience about oncology, that can make sense of this data, and that has been trained, if I talk about AI, on the clinical literature, on the clinical experience that we have as clinicians, on the guidelines, on the databases. And all of this we want to be able to integrate, and of course machine learning should be a tool that we want to be able to exploit for this. But modeling, once again, I think is very important because we want to be able to simulate at one point. Of course, what I show you here is more a wish list. I don't know, I hope we can do this, but we have to push towards this. We have to collect a lot of data to be able to model, to simulate, and as physicians, we really want to be able to simulate. We don't want anymore to test in real a treatment that will not work and pass to another treatment. We want to simulate so that we can give a toxic, ineffective treatment in a virtual world, not on the real patient, and be able to avoid this on a real patient. We want to be able to have a digital twin that is continuously updated, because as I showed you, cancer is evolving. I mean, a patient evolves. You don't stay the same. Each day you change. We want to be able, if we develop a tool, to validate it. The question of validation is really important. We really want to be able to do this to predict the best treatment for our patients. So this is the official slide, I think, of the project that you may have already seen. 1 minute on the challenges. We can talk hours about all these challenges. How do you validate a digital twin? I told you, probably do a clinical trial. But the tool should be evolving. This is problematic for regulatory agencies. We have to change that. This is all about the stuff about AI tools in clinical practice. This is a very this is a nightmare also, but we have to advance towards this. How do you represent cancer? I talked about modeling, but how do you represent mathematically all of these scales? This is a very important question. How do you represent it to a patient? How do you collect enough training data to model, to fit the parameters? This needs a lot of money, a lot of patience, a lot of biopsies. How do you standardize all of the data? How do you develop the infrastructure? Who will govern this? Who will own the data? This is patient data. Will physicians accept these kinds of tools if they don't understand it? And of course, then you have to educate physicians, but also patients for these tools. What we want to do as oncologists is be able, one day maybe, to do a consultation with the patient and with its digital twin, test treatments on the digital twin and say to the patient, okay, you see, we think this is the best treatment because these are the ones, we tested it in the virtual world, it doesn't work, it's too toxic. Want to have synthetic data for clinical trials. We even want, I don't know if we are able to, but is AI able to give us insight into biology, make biological discoveries? This is even a philosophical question that we ask ourselves as biologists and clinicians. To finish, I would like to acknowledge, of course, all the people in Gustave Roussy, especially Fabrice André, my boss, who is the chief, I would say, on MediTwin, and Nicolas, and all the people in Dassault Systèmes that work with us.